right? It's very low frequency. This is this portion of the vocal repertoire indicated here. Look at the difference in the labia. Not surprising again, it's exactly what you would expect. But seeing is believing, yes? And then we looked at all 8 species that we analyzed this way, and we wanted to know whether the frequency ranges in the vocal repertoires are somehow correlated, A, with the asymmetry between left and right sound sources. And we find a positive correlation for asymmetry in the lateral labia and the medial labia. And then we wanted to ask if we detect layers, and we just, somehow arbitrarily determine this is a layer and this isn't. But the more layers a bird seems to have in terms of arrangement of collagen and elastin, the larger the frequency range of its vocal repertoire. So here I think we have a situation where we have a morphological structure that facilitates the production of a broad frequency range. And then neural control in this boundary, within these boundaries, will act and make this species-specific vocal repertoires. And then I told you about the pulse tone, which is sort of a cheap way for a bird to fake low-frequency sounds, even if they are small. Okay, and my final story now is shifting gears a bit. I'm going to tell you a story about what birds dream of in regard to song at night, how we know this, and then I'm going to propose a hypothesis that is blasphemy. I like to push buttons, and I surely do. So it goes to everything people want to accept. But we'll see. Maybe I'm happy to hear your insights into this. Okay. So I will start out by claiming, and Jon and Sarah will immediately cringe, that song is very stereotyped, since they study plasticity. It's still stereotyped, but there is plasticity, okay? It's very stereotyped, and in some species, the stereotypy occurs every breeding season, like in the starling, and then when testosterone levels drop, males get smart again, they can learn new things. So starlings can add to their vocal repertoire every off-season, and then during the breeding season they are stereotyped like crazy. But other species, like the zebra finch, they fix a stereotyped song at the end of vocal learning at about 3 months of age, and then this stereotyped song is sung for the next 4, 5, 6 years, pretty much unaltered, unless experimenters mess with them. Okay? Now, I'm calling it high stereotypy, but certainly for a long time, and I will explain what I mean so Jon and Sarah do not have to immediately protest, right? Because it's not for sure. This is this song. Okay, you detect pressure patterns. This is the sound. This is the spectrogram. You hear the repetition of 3 to 5 syllables, which is typical for zebra finches, over and over and over. Okay, all his adult life. And if we expand this little section, you sort of see the details, different syllables. Each syllable has a characteristic pressure pattern that gives rise to it. Okay, so even the pressure is very stereotyped, of course, so it doesn't generate the same sound with different motor patterns. And if we looked at an EMG, just any arbitrary EMG we measured in this bird, it would look like this, and sound like this. Also stereotyped, yes? If we look at the upper vocal tract movements, the beak movement, fairly stereotyped. If we look at this expansion of the oropharyngeal cavity through hyoid movement, also stereotyped. So every aspect of the motor pattern of producing this song is highly stereotyped. How stereotyped it is there's about a 3 to 5% variation in duration. So zebra finches make a difference whether they sing by themselves or directed to a female. When directed to a female, they sing faster. I wonder why. And people have looked at zebra finch song across the whole day and found about 100 hertz variation in fundamental frequency over this time period. We already know that pressure can affect frequency, right? So if they sing more and less loudly over that period, that would explain part of this variation, perhaps. And furthermore, Katharina Riebel and her group, they have studied female zebra finches and asked the question whether females pay attention to stereotypy. And it seems that stereotypy was very important to females. So I would argue then, if they make such small errors only, that you actually get very little in terms of message from your peripheral feedback mechanisms. There's very little acoustic feedback error, and certainly very little somatosensory feedback. And why I'm pointing this out is because it's drastically different during vocal development, where we have the sensory motor learning phase, and from then on 90 days in zebrafinch, we have stereotyped song, we call it crystallized song. But if you, that's a practice in zebrafinch, right? Babbling. Only mother will understand what the kid is saying. And then the same bird as an adult. Okay? So during the practice phase, they make lots of errors. And we think that feedback allows them to fine-tune their motor skills to imitating the song they had memorized at one point during development. Okay, so we'll look a little bit about what birds do in their sleep, and then I'm going to propose the blasphemy hypothesis that that this allows the birds to maintain this stereotype song. And let's see how successful that is. Okay? So we know that neural activity occurs during sleep in pretty much every mammal that's been looked at, and probably, obviously, as you know from your dreams, vivid dreams in you too. And we knew it in birds. And the idea was that this neural activity is very important to consolidate memories. And there have been plenty of studies where a little task was taught to a subject, and some got to take a power nap and others didn't, and the ones with the power nap did better afterwards. Okay, so memory consolidation is certainly something where we have good evidence that sleep might help with that. And this also extends to motor skills, motor memory, sensory motor activity. Okay? And in mice, we have very good evidence that if you let them run in a maze and they have these place cells in the hippocampus that fire at particular points, they seem to replay this in their sleep, and if they get to do this, they remember the location better. So that's this hippocampal one. We also have evidence from these forebrain areas in songbirds that neurons seem to be active spontaneously, as if they were singing. But certainly, when you play back the song to the sleeping bird, you see song-like activity repeat. Okay? But what the bird, that one neuron is doing, is hard to tell. It's only one neuron in the big number that make up a motor group. So we don't actually know what the bird replays in its sleep. We just know it's song-like. And we made the fortunate discovery that when birds do this in their sleep, they activate the syringeal muscles. Okay? We were measuring EMGs and syringeal air sac pressure to study the role of syringeal muscles in frequency control. But one undergraduate student said, "Hey, can we do this at night? I want to do playback." I said, "Sure." And sure enough, the syringeal muscles are activated at night, and also spontaneously. And then we started studying this systematically. Okay, so here is the song of a bird, where we have two muscles with the EMGs, the air sac pressure, and two motifs repeated. And I just want to tell you that these muscle patterns are fairly stereotyped and characteristic of syllables. So by looking at EMG, I can tell which syllable the bird is singing. Okay, so that's step number one. And then we record all night, and we have an algorithm that uses the EMG pattern and looks through the nighttime recordings. Okay, and so here you have in different colors the different syllables of this bird and some burst of nighttime activity that actually lines up perfectly with every syllable in this EMG set. Okay, so now we can look through weeks of recording at night and figure out what does the bird sing in its sleep without singing, because it is without singing. We have song here and a nighttime activity here where you see the characteristic patterns, but look at respiration. The bird's not pushing air past its syrinx. It's breathing the same way as it did before. Okay, so it's not increasing pressure. Look at the scale, 0 to 1 here, it's 0.1, right? So it's not increasing its pressure, but it's, and that's indicated here, where we have duration of a respiratory phase and the amplitude of the pressure. This is during song. Okay, this was a bird that came close once, so we pulled it out just for interest's sake. But it's not happening at the moment. So now we look at what the bird is singing during the night, fictively singing, and we line it up, and you have some where the left and the right side sing all syllables of the motif. Then you have another one where the left side sings the green syllable, but the right side omits it, and you can go through and see all kinds of aberrations. One that is cute to point out is on this left side, the bird skips one syllable and sings the next, whereas the right side sings the normal motif. So syllable 4 pops up here, while on the right side the bird sings syllable 3. Okay? And short segments, individual syllables, everything you can imagine the bird seems to be doing. Half syllables, where only half the pattern of a syllable appears in the nighttime record. So, another interesting thing, it seems to be sort of rhythmic. The bird isn't dreaming at a particular time of the night or morning, but it seems to come in rhythmic bursts throughout the night, and that probably is correlated with sleep phases of birds. I once did metabolic studies, and I find exactly this type of rhythm in metabolism. We haven't done EEGs to confirm sleep state. But the other aspect of this graph is that you see different templates. So this is one syllable, and then several other syllables, and you see that they are practiced with different frequency throughout the night. And the whole sequence appears very infrequent. 8% of the cases is the full motif. Everything else is some kind of variation. Okay? So very infrequently does the bird sing the whole motif. So it doesn't produce sound because respiration isn't engaged. It practices all kinds of variations. Now, remember, the way we find these is by using song-like EMGs. The next step in our analysis is that we look through for song-like activation that doesn't match anything. So we may find even more diverse patterns. And certainly there's uncoordinated activity between left and right. So this is all not practicing stereotyped song, I would argue. Right? 8%, that's 92% is not. And referees say they should look at the 8%, because that's where the actual practice happens. Okay. I'm not quite convinced. At any rate, so all this nighttime activity is called replay, and I'm asking, is this a fortunate term? Is it replay? What could it be doing? Well, I'm proposing that it generates internal error signals that the brain can use. So I would even go as far as, way out on the limb, suggesting that it may actually recapitulate developmental processes or the big errors it generates during development during these nighttime phases. And that I would claim, without having a mechanism, which is a very big shortcoming of my hypothesis, that this could allow a zebrafinch to maintain a stereotyped song without making many errors over 4 to 6 years of its life. Something most of you may know or may not know is in these song control areas, new neurons are incorporated, more so at the younger adult age than the later adult age, but they're incorporated. These neurons need to be going into a circuit and doing the job right. If you have to sing a stereotyped song, how do you get this right on first try? Again, my suggestion is nighttime activity may do something. And I'll go through this quickly. We were puzzled, why activate the syringeal muscles? That makes no sense, right? Does it have a function? And so one possible thought is that in order to get the internal, within the brain, circuitry in the right frame of mind, if you will, you have to activate the motor neurons of the syrinx, because they are in the brain, right, in the hindbrain, and hypoglossal nucleus. And respiration, we have the premotor circuit in the hindbrain. So the motor neurons, of course, sit in the spinal cord, and maybe there's an inhibition, because it probably isn't very advantageous to sing all night if predators are looming. So that's one possibility. We have no evidence that any of this is true. Maybe because they are not making sound and there's no external movement, there's no selective pressure to cut it out. That, of course, leaves us with the question, well, activating muscles is costly. To some degree, can they afford this energy expense? No answer to this either. And then we were thinking, from looking at the super fast muscles of the syrinx after a nerve cut, within a day and a half, the super fast fibers disappear at a high rate from these muscles. So the myosin turnover is very rapid in these muscles. And so we were thinking, oh, wow, maybe 8 hours of sleep is too much. You should activate them so myosin expression does not get altered. We find no changes after 8 hours or 10 hours, so I don't know, and this is probably barking up another wrong tree, but nevertheless it's been a lot of fun, and this is, I think, just the beginning, because there's so many questions. You know, we can now ask in the brain, if we interrupt the circuitry here or there, how does that affect song in the long run, etc., etc. So the mechanisms of this would be very interesting to explore, and all these other birds that learn every off-season. When do they dream about singing? Or do they? Nobody knows. So in summary then of the whole thing, I hope to have convinced you that the morphological diversity of the syrinx probably has a functional role, and that some of these simple syringes of suboxynes may actually be more sophisticated in their morphology than thought about tracheophones. How neural control and morphological structure integrated to control frequency in oxynes is a story that I think is interesting to me, because we've moved away from just neural control. Right now we are actually looking at the hardware piece as well. And to what degree vocal learning is responsible for these differences is an interesting question. So have Austin specialized on software, if you will, somebody gave me this idea today, I'm stealing it, versus hardware changes, and have some Austins invested in quotes, in the hardware changes because they don't have learned vocal behavior. And then this story about stereotypes. Of course, I've been fortunate to work with a lot of people. This is not an exclusive list, but the people with little stars next to them are the ones whose work I've shown here in more detail than others. But it's been great both working with colleagues and especially great to learn from my students, because whatever you may think, it does go both ways. Thank you.
Yeah, so this was very nice, very interesting. The error signal then is not auditory.
That's correct.
It's not auditory feedback. So what
So
pathway, or is it, I mean, the mechanosensory stuff could still be going on.
Yes, so in fact all the somatosensory information must be weird, right? Whatever is coming back from the periphery ought to be quite misleading. There's no auditory feedback. Respiration isn't engaged, but whatever feedback we get from the syrinx shows movement. So we don't know anything about muscle spindles and syringeal muscles. Nobody's been able to find them. I don't know whether it's because people haven't looked super hard, which is true, they haven't. But in every other muscle, you look at it and you see them, right? So I think they're not there. Whether they're free nerve ending sensors, nobody knows. We know there's afferent information in the tracheosyringeal nerve, but yeah, very little is known. So that's one problem. Peripheral feedback is not going to be much use. But in the brain, there may be an efference copy, and that efference copy would be useful in the internal process. And so that's what I'm thinking may be a benefit here, that you can use the efference copy without actually generating the sound. But who knows? It's speculative.
Great, so yeah, that's fascinating stuff. So I was worrying about this as well. So no auditory feedback, and so either there's somatosensory feedback, arguably that even if there aren't muscle spindles, there's probably cutaneous afferents of some kind.
Sure.
But so then when you have to have if you're going to make use of cutaneous information or even in efference copy, you have to have some goal against which you're going to compare it if you're going to use this to drive learning. So that's
yes.
where
So my thinking is peripheral feedback is probably not even getting there. The efference copy is helpful because they already have a stereotype song.
Oh, so this is variation in relation to that. Okay. Awesome.
Okay, so then of course that asks an important question that I haven't phrased yet. What do young birds do at night, right? When they actually still practice. They wouldn't have that yet. And so we need to look. I think that would be good.
Can I just ask one other quick question that I have to run? So how much, in humans, the upper vocal tract contributes enormously to the characteristics of speech and sounds. And so to what extent is this true in the birds that you looked at? So for instance, if you pin the beak,
yes.
so it can't move, then what changes does it have to the quality of sound?
It changes the upper harmonic content. So if the beak is in a closed position, that's used for low frequency sounds. If you keep it closed for high frequency sounds, it comes out soft. We don't know whether it's linked mechanically or neurally to the resonating chamber, the OEC. So that hasn't been looked at. It could have an indirect effect, therefore. But yes, it does have an effect. But interestingly, when we manipulate, like you would manipulate upper vocal tract structures, right? Humans pay attention. We've manipulated both the OEC and the beak, and the bird doesn't seem to change its motor pattern. Now, how long we would have to wait, I don't know, but not for a week or two. So there's no instantaneous correction that we've seen so
That's
far.
curious.
Yeah.
Quick question. So,
Yeah.
so is the idea that if you injected random current into the series during one bird's sleep, that you would lead to a gradual deterioration as well? If this is somehow important for plasticity or maintenance as well? If you just
Then I'd say that will only be if peripheral feedback is important. But I'm claiming that probably it's not. I'm claiming that, proposing, without any evidence, that this is all happening in the brain.
Because the search I did before was just to look for the template of the spiking pattern, and they didn't report, like, so Dan Margolius' paper, they didn't report the false rates, right? Just try to look for that template of spiking, motif completion.
Sure.
So you're saying you would look for motif incompletion in that data set, you would find an abundance of incomplete motifs.
Well, I would say if you could detect it by looking at one neuron, but how could you, right?
In terms
A
of,
syllable is generated by contributions from many neurons, so since we can only record from a few at a time, we will never know this from neural recordings, is my claim. Dan may disagree. He would disagree.
I was wondering if you can get some clues about what's happening because you have this left, or the different signs have, you said that they could sometimes be quite different and one would skip a syllable and one would not, but they didn't look like they were completely random. They seemed to start on the same syllable. And so, I mean, do we understand enough about processing further up to understand sort of where things might be diverging then?
Yeah, the synchronization of the left and right sides, since from very high up it's all ipsilateral. Correct me if I'm wrong, but I don't think we understand it very well. So I don't even know where to start. We don't know exactly how the two HBC areas are synchronized. So there must be a signal from somewhere else. And people argue where it's coming from, if it's at all coming from somewhere, which it always has to. I don't think we know it.
So going back to John's question about messing up the periphery basically, and then you said, well, but if it's an efference copy thing, something internal, so you could start looking for where that might be. I mean, these neurons come from RA, right?
Yes.
So you could start trying, well, maybe the efference copy comes from RA, so you could at night mess up pharmacologically or so, firing patterns.
That's what we're trying to do.
Oh,
next.
okay.
Yeah.
Good.
And that's been well described.
I thought I had it.
Models in mind, right? Other people have proposed this, but that's what we're trying to do.
Mhm.
It's just you have to, or we're trying to paralyze these areas for 8 hours. It's not trivial. Yeah. And then in the morning it has to work, right?
Yeah.
okay, well, we have some refreshments and some snacks outside. Also, we made a little bit of snafu since it's our first seminar of the year. There's no wine this time, but we were going to take Franz over to the Thompson House in about 20 minutes, so equal fast. And then see you over at the Thompson House. Anyways, thank you, Franz.
Thank you.